Hello everyone. Today I want to introduce you about the rapid and widespread evolution of bioluminescence in the marine fishes. And I hope after this talk you can be have more interesting about the evolution of the fishes. So first I need to briefly introduce you about what is the bioluminescence. Bioluminescence is the production and emission of light created from the living organism, and the light is created by the chemical reaction between the substance luciferase and the enzyme luciferase. And this picture is the bioluminescent creature in the sea, the lantern shark, the bobtail squid, jellyfish, and the shrimps. And typically, the bioluminescence can be separated to two types. The first one is the intrinsic bioluminescence, means that the creature made the light by themselves. And the example is the midshipman. And this is the bioluminescent pattern of it. And the second type is the bacterially mediated bioluminescence, means that the creature create the bioluminescent light by the bacteria symbiosis. So they are not create the light by themselves, but by the bacteria. So the example is the fleshlight fish. So the bioluminescent organ below the eyes, you can see there's the pattern of the bioluminescence. And what is the function? What is the function of bioluminescence in the ocean? The first one is the defense. We can see this fish emit the bioluminescent liquid to create the smoke, and liquids misdirect its predator and make it have the time to escape, just like the ink in the squid or octopus. And the second, the camouflage. We can see this picture. There has the bobtail squid has the bacteria symbiosis bioluminescence in the ventral side of the body. So the lizardfish is predator sees from below the squid. The light will mimicking the light penetrated from the sea surface. So the lizardfish can see it upward, and this kind of camouflage called the counter illumination. And the third one, I think this is the most famous example, the deep sea anglerfish. This specialized dorsal fin called the S-glass can have the bioluminescence, can attract the prey to make this prediction in the deep sea more easier. So the first function of bioluminescence is the communication. In the picture, we can see the different sexual, the male and the female has the different bioluminescent pattern. That means that this pattern can make them to make attraction or recognition within the same species. And the end of this study is they want to determine the number of independent evolution original of the bioluminescence. Means they want to know how many times of the fish has evolved the bioluminescence. And the second is they want to identify which clade has a bioluminescence within the red fin fishes. So what is the red fin fishes? So the red, it means the bony structure supporting the fish fin. And the red fin fish comprise nearly 99% of the extant species of fish and over 50% of living vertebrates. So most of the fish species is the red fin fishes. So the salmon are red fin fish, but the shark, the sea can, and the lampreys, they are not red fin fishes. And the third, the end of the study, they want to test whether the bioluminescence plays a vital role in the diversification within the ocean. So the material amassed in this study, they used the 10 nucleotide plus 1 mitochondrial gene to reconstruct phylogenetic tree. And the phylogenetic tree is the hypothesis tree to show the relationship between the different species, like this tree. And in this tree, the blue one is the species present of the bioluminescence, and the green means the species extent of bioluminescence. And secondly, use the fossil to corroborate the relative division times. Means that they use the fossil record to know the ancestor of the past fish, to know when the two species to separate it from the common ancestor. And in this tree is a time tree, so each branch lines means the time there from the Devonian to the Cenozoic. And the third, they use the Bayesian ancestral character state reconstruction. And in the tree, the purple bar means the node, means the most recent common ancestor. And each node, by the Bayesian way, we can know whether each node has the bioluminescence. This character have appeared in their ancestor. And fourthly, use the software Medusa to estimate the net diversification rate and the relative rate of extinction. So how to read the phylogenetic tree? So I take the Bericiformes as an example. So first, we can know that the most recent common ancestor of the Bericiformes appeared in the late Cretaceous, from here to here. Second, the bioluminescence appeared in the Bericiformes during the early Cenozoic. We can see the node 23 is about 50 million years ago. So the last one, we can see the node 23. If the bar has the more black, means that the bioluminescence is extended in this ancestor. And like the node 24, if the bar show more blue color, means that the bioluminescence has present in this ancestor. So the result, the evolution of bioluminescence across the red fin fish. So we can see the first evolved from the early Cretaceous about 150 million years ago in the Stomiformes. And at least 27 independent evolution events across the 40 major red fin fish language. So each node we can know whether the bioluminescence is independent events. And the third, the inhabitat of the bioluminescent fish is ranging widely from the inshore coral reef to the deep sea environment. And for the evolution of the intrinsic bioluminescence, it happened at least 8 times, and including more than half known bioluminescent fishes. And have two most species-rich languages, the dragonfish family Stomiformes, including 426 species, and the Metyphoformes, the lanternfish family order, including the 256 species. And the intrinsic bioluminescent fishes show the higher species richness than other clay. And this curve pattern is constructed by the 95% confidence interval of this parameter, the net diversification rate and the relative rate of extension. And the unit is the language per million years. And in this figure, the x-axis is the their age per the million years. And the y-axis is the number of species. So we can see the dragonfish, lanternfish is much more higher species than the general curve. And for the bacterially mediated bioluminescence, it is 17 times, and particularly among the former the spiny red fin fishes. So the spiny red fishes is for the example is the golden diamond pineapple fish. We can see its dorsal fin has a spine. Is the character of the spiny red fishes. And the bacterially mediated bioluminescence still high reaches in the young clay. And the short summary is the bioluminescence first appeared in the Cretaceous in the Stomiformes, and at least 27 times in the red fin fish, 8 times in the intrinsic, and 17 times in the bacterially mediated. And both intrinsic and the bacterially mediated bioluminescence show high species richness. And how bioluminescence enhances the fish diversity. So one of the important functions is the communication for the species-specific use. And this species-specific use can improve promoting the sympatric speciation in the fish population, because the different sexual have a different pattern, so they know that their own species has their own pattern, so they can recognize them.